Hi everyone. Hope you're all doing good. Welcome to yet another episode of our season two's RRR. That's Read, Revise, and Repeat. And today's session, we're going to be discussing genetics for your part one exam. And I hope it's going to be beneficial. So, for the quote of the day, I've related it to genetics. Now, your determination is like a perfectly sequenced gene. It drives everything forward. And just as every tiny nucleotide plays a role in creating something extraordinary, every moment of effort that you've put in will shape your success. You must trust the process and you're destined to evolve into excellence. So, the agenda or my aim for today would be to clear a few concepts that you might all have doubts in. Now, genetics on the whole is a it's a high yield topic and a very fascinating one actually for the exams. Understanding key principles such as inheritance patterns, gene mutations, their implications can actually help you clear this exam easily. Now, this is a topic where clarity and logic will prevail. So, you have to actually have clear concepts, you have to grasp the basics, and the rest falls into place. So, that will be our agenda for the day, clarifying concepts so that you're exam ready and you can ace genetics. Now, let's begin with one of the most interesting concepts, and I must confess, before I started my journey for MRCOG Part 1, I really wouldn't have been even aware of this. So here goes our first question. Let's just go through it and discuss about it. There's a 32-year-old woman who presents to the clinic for genetic counseling due to a family history of mitochondrial myopathy. She is concerned about the inheritance pattern and asks about the genetic makeup of mitochondrial DNA. Which of the following best represents the number of genes present in the mitochondrial DNA, whether it's 20, 37, 46, 100 or 120? So I'm sure you must have come across this question a lot of times, and by now you would be pretty aware about what is mitochondrial inheritance. Now, the mitochondrial genome compared to the nuclear genome, mitochondrial genome is built of around 16,569 DNA pairs, whereas the nuclear genome has around 3.3 billion DNA base pairs. Now, these base pairs which are there in the mitochondria are essential for normal mitochondrial function, and they provide instructions for enzymes involved in oxidative phosphorylation. Now, unlike the nuclear genes, which are inherited from both parents, mitochondrial genes are inherited only from the mother. And do we know why? Now, let's try and understand the concept a little bit. So, this is what is a spermatozoon, and it has three parts: the head and the tail. The tail is made up of the midpiece and the principal piece, and it's the midpiece in which the mitochondria of the sperms are located. And these mitochondria are actually very essential for sperm function, like the movement, capacitation, acrosome reaction, and also the sperm and oocyte fusion. However, at the time of fertilization, it's only the head which enters the ovum. The tail, which comprises the midpiece and the principal piece, is not a part of the zygote that is formed. Therefore, the mitochondria that the zygote will develop from, the genes, are from the mother only. Okay, so this, unlike the nuclear genes, okay, which are inherited from both the parents, these mitochondrial genes are inherited only from the mother. So if there is a mutation in the maternal mitochondrial gene, then it is passed on to all the children. Okay, so the mother will pass on to all the children, And if suppose she has a son, the son will not pass it on, but the daughters will again pass it on to all of their children, and so on. Okay, so this is known as a maternal inheritance pattern. And so now let's look at the answer. The mitochondrial DNA has 37 genes. Let's have a look at our second question. A 45-year-old woman with a strong family history of breast and ovarian cancer undergoes genetic testing which reveals a BRCA1 mutation. She asks about the type of ovarian cancer most commonly associated with this mutation and which of the following is the most likely type of ovarian cancer in this case. Options given are A. Mucinous carcinoma, B. Clear cell carcinoma, C. Endometrioid carcinoma, D. Serous carcinoma, or E. Germ cell tumor. Alright, so the answer over here is serous carcinoma. Now we all know BRCA1 and BRCA2 mutations. These are strongly associated with high-grade serous carcinoma of the ovary, which is the most common subtype linked to hereditary ovarian cancer. Now these mutations in these genes impair DNA repair mechanisms, leading to genomic instability and increased cancer risks. High-grade serous carcinoma is an aggressive type of epithelial ovarian cancer. Now, BRCA1 and BRCA2 are genes that are involved in DNA repairs. However, there are certain pathogenic germline mutations that can happen in these genes, and these are associated with hereditary carcinomas, most common of which are, of course, breast and ovarian. However, they are also associated with other cancers like male breast cancer, pancreatic cancer, and prostate cancer. Now, there's a lot of confusion amongst all of us actually regarding the numbers that might be quoted. So this chart is from the NHS Genomic Education Department, and it was released in 2023. So I believe that we can trust this and take a note of these numbers, and in case the exam question asks, then we can rely on these numbers and then give our answers. So this is another question which is actually a direct question, but if you know kind of a question. So a 6-year-old child presents with signs of ambiguous genitalia and salt wasting crisis. Genetic testing confirms a diagnosis of congenital adrenal hyperplasia due to 21-hydroxylase deficiency. On which chromosome is the gene responsible for this condition located? Is it chromosome 5, chromosome 6, chromosome 7, 11, or 17? Now let's move and see what the answer is. So the gene which is responsible for congenital adrenal hyperplasia, CAH, specifically CYP21A2, is actually located on chromosome 6. So our answer is B, chromosome 6. Now this is within the major histocompatibility complex and that region which is also on chromosome 6. Now mutations in this gene lead to enzyme deficiencies, and most commonly it is 21-hydroxylase deficiency, which disrupts the cortisol and aldosterone synthesis. Okay, and then the disease that gives rise to is congenital adrenal hyperplasia. So these are conditions which are important in the context of gynecological genetics, and knowing the associated genes and chromosomal locations can actually aid in understanding their inheritance patterns, clinical manifestations, and potential genetic counseling. Moving on to a related question. Which type of endometrial carcinoma is most likely associated with mutations in the mismatch repair genes? Whether it is type 1 endometrial carcinoma, type 2 endometrial carcinoma associated with Lynch syndrome, clear cell carcinoma, or endometrial stromal sarcoma. Now, what are mismatch repair genes? Now, mismatch repair genes, these are a group of genes which play a crucial role in actually maintaining the integrity of the DNA by correcting any errors that can occur during DNA replication. Now, these errors such as if there is any base pair mismatch or any small insertion deletion loops which can happen. So if they arise during DNA replication, then these mismatch repair genes kick in and repair this before the DNA proceeds for replication. But if there are mutations in these genes, that condition is strongly associated with carcinomas, and one of the main significant hereditary condition that it is associated with is Lynch syndrome. Okay, so these tumors, they actually present at an earlier age and are often of the endometrioid or type 1 endometrioid type, but the association with Lynch syndrome is key to identifying the genetic cause. So I guess now you all know the answer. So yes, it is endometrioid carcinoma associated with Lynch syndrome. These are mutations or defects caused due to the mutations or defects in these mismatch repair genes. So the body loses its ability to correct errors in these DNA sequences, and this leads to accumulation of mutations and increasing the risk of developing cancer. This syndrome or this condition is Lynch syndrome. It is also called hereditary non-polyposis colorectal cancer or HNPCC, where individuals are predisposed to several types of cancer, including colorectal, endometrial, ovarian, and also some other types of cancers. Okay, so this is another question that there's a lot of confusion about, and I have put it here so that the confusion is now finally sorted. A 32-year-old pregnant woman at 16 weeks of gestation is considering amniocentesis for genetic testing. She is healthy and has no history of complications, but she is concerned about the risks associated with the procedure. What is the additional risk of miscarriage associated with amniocentesis? Is it 0.1%, 0.5%, 1%, 2%, or 5%? Now let's see what the answer is. So the answer is 0.5%, and this is the reference from the website, which says, and these are the points that I want you all to note, that in a singleton pregnancy, if there is amniocentesis or chorionic villus sampling, both of these, then the risk is actually, if it is performed by a skilled operator, it is likely to be below 0.5%. That is there in the first line. And then in case of twin pregnancy, following chorionic villus sampling or amniocentesis, performed again by a skilled operator, the risk is around 1%. Okay, so depending upon what the question asks, your answer will vary. It is 0.5% in a singleton pregnancy and 1% in a multiple pregnancy. Now this really is one of my favorite parts of genetics, and we'll see why. Okay, so here is a pedigree chart and you are supposed to identify the mode of inheritance. So have a look at this pedigree chart. Maybe you can pause this video here and try and reach your conclusion, whether it's a autosomal dominant, X-linked dominant, autosomal recessive, X-linked recessive, or a mitochondrial inheritance pattern, and then move on to the next slide and we'll see what the answer is. Alright, so this here is an algorithm, okay, which consists of 4 main questions that we ask ourselves when we see a pedigree chart, and depending upon the answers, we will move according to it and reach a conclusion as to what we are dealt with, okay, what we have been handed over. So the first question that we ask ourselves is, are only males affected in the pedigree that is given to us? So the first question itself is a negative, so we are not dealing with a Y-linked inheritance. We move on according to the algorithm. The next question that we ask is, are there any cases where an affected child does not have an affected parent? So here we have an affected child and the parent is not affected. So yes, we are dealing with a recessive condition here. Now we have to ask whether all the sons of an affected mother are affected or are more males affected. So here we see that all the sons in this pedigree that has been given to us of an affected mother are affected. So here we are dealing with, what are we dealing with? Let's see. So this is an X-linked recessive pedigree chart. Now we have a Kickstart series that we had recorded last year on pedigree charts, in which I've explained this algorithm a little bit more in detail. So I would want that if you have time, please go and have a look at that video also. So the flowchart becomes a little easier to grasp, and I hope this will really help you in solving the pedigree charts, which all of you are really afraid of, and you will not make a mistake in them. So this is also a very often repeated question. Maybe I've a little bit skewed it a little bit. So let's see what the question reads. A researcher in a genetics lab is working on gene editing to correct a genetic mutation causing a rare inherited disorder. The lab is utilizing CRISPR-Cas9 technology to introduce precise changes to the DNA sequence. The researcher is familiar with how this technology works and the potential applications in the various fields, including medicine and agriculture. What is the primary function of CRISPR-Cas9 in gene editing? It is a combination of DNA and enzymes and helps to cut RNA and inhibit translation. B. It is a combination of RNA and proteins and helps to introduce double-strand breaks at specific locations in the DNA. C. It is a combination of RNA and proteins and helps replicate DNA during cell division. D. It enhances expression of genes. Or E. It helps create random mutations in the genome. Now, I hope you all know by now what CRISPR-Cas9 is. And let's see in the next slide what the answer is, and let's discuss about this. So, well, we must know that Cas9 is actually an enzyme, and CRISPR is actually a short form of clustered, regularly interspaced, short palindromic repeats, and this is actually an RNA component, okay, which is actually a guide RNA component of this combination. So amongst the options that have been given, option B, which mentions that it is a combination of RNA and proteins, which is correct, and this helps to introduce double strand breaks at specific locations in the DNA. So this is our right answer. Now what actually this CRISPR-Cas9 is actually like a, you can say it is like a cut and paste tool in the hands of the scientists who have developed this CRISPR-Cas9. Now, the CRISPR is a guide RNA, okay, and that is actually kind of linked, or it is actually made to link with the DNA of the responsible cell, okay, which is causing some disease or some problems, and it is introduced with this Cas9 protein into that cell, and it will go there and then attach to the DNA, and using this technology the scientists actually clip the responsible segment. They can either insert some new sequences there, or they can delete that segment completely and repair that defect in the DNA which is happening. So together this RNA and protein forms a precise system for editing any abnormal DNA that might be present. So the RNA identifies and binds to the target DNA, and the Cas9 protein that will cut the DNA at the specific location, and the cell's natural mechanism will then be kicked in and it will fix the break, allowing for gene editing, whether it is insertions or deletions or even modifying genetic material. Now, one of the most asked questions, okay, this one should not go wrong in. A 34-year-old woman is admitted to the labor ward via the emergency. She has not had any contact with health services during her antenatal period. Emergency cesarean section is performed due to signs of fetal distress. a small female baby is born and noted to have cleft lip and palate, prominent occiput, low set ears, omphalocele and clenched hands with overriding fingers. The baby is transferred to SCUBU due to apneic episodes. What is the most likely diagnosis? So we are dealing with an aneuploidy here, and we should be aware of the signs and symptoms that the most common aneuploidies, especially Down syndrome, Patau syndrome, and Edwards syndrome, Cry-Duchat syndrome, these will present with, and then we should be able to answer this question. So the options are trisomy 13, trisomy 18, trisomy 21, Turner syndrome, or Cry-Duchat. Now let's move on to our next slide where I've detailed it a little bit and proceed to the answer. Okay, so this slide is actually showing you the most commonly asked aneuploidies and the most commonly occurring also, and the features that they present with. So we must know that these are the features which they are describing in the vignettes, or and be able to answer as to which kind of aneuploidy are the symptoms referring to. So now the features that have been described in the question, actually they are seen in both trisomy 13 and trisomy 18. Now what happens is a few of them are also in Cry-Duchat. Now why will we go for one answer over the other is because Edwards syndrome, that is trisomy 18, is more common than Patau syndrome. Now we all know we are dealing with single best answers, so we need to choose between which is more likely to happen. So since the incidence of Edwards is more common, therefore our answer here will be option B, which is trisomy 18, which is Edwards syndrome. So here we are, our answer is trisomy 18. Another very interesting question. Actually, genetics is actually very beautiful. So very interesting because you have to use a lot of logic, like I said. So now, given that humans have 23 pairs of chromosomes in a dividing cell during metaphase of meiosis 1, how many telomeres are present? Okay, so what are telomeres? First question that you should ask us. What are telomeres? They are present at the end of each chromatid. Okay, so each chromatid has 2 telomeres at each end. So now the options given are 23, 46, 69, 92 or 184. We have been asked about metaphase of meiosis 1. Now we should know the sequence also. We have prophase, metaphase, anaphase, telophase. Till anaphase, the chromosomes, the sister chromosomes are linked to each other. Okay, so remember that. Let's proceed to the next slide and see what our answer will be. So now let's take it step by step. Now we know that humans have 23 pairs of chromosomes. In the S phase, the DNA replicates. Okay, so each chromosome will now get 2 chromatids. Now each chromosome consists of 2 chromatids during meiosis 1. Each chromatid has telomeres at each end. So each chromatid has 2 telomeres at each end. So now we calculate there are 46 chromosomes, which means there are 92 chromatids, and each chromatid has telomeres at both ends. So we have 184 telomeres. And let's see what the answer will be. The answer is 184 telomeres. So if they ask you how many chromatids are there, that's how you proceed. And if they ask you in anaphase, then you will obviously have to halve it. Another very frequently asked question, Robertsonian translocation usually involves which of the following chromosomes? 13, 14, 15, 21 or 22, 18, 21 or chromosome X, 6, 17, 21, X and Y, 1, 6, 13, 14 and 21, 13, 18 and 21. So our answer is option A, 13, 14, 15, 21 and 22. Now we must know what are Robertsonian translocations and why are these particular chromosomes involved. So Robertsonian translocations are actually the most common recurrent constitutional chromosomal aberrations in the humans, and they have an incidence of around 1 in 1000 individuals. They are most often formed by centric or short arm fusion between two different acrocentric chromosomes. So that is why it is chromosome 13, 14, 15, 21 and 22, because these are acrocentric chromosomes. Now, what are acrocentric chromosomes? I'll be talking about shortly. But what happens in these is that you know about the P arm, which is the short arm, and the Q arm, which is the long arm. So there is a breakage, and the two short arms of these acrocentric chromosomes, they combine together, and the long arms of these chromosomes combine together. Now, these two short arms, which are usually very insignificant, they are lost, and only these two long arms of two different chromosomes, they are present in those individuals. And this kind of translocation between two acrocentric chromosomes is called Robertsonian translocation, and it is very commonly seen in Down's syndrome. So these are the types of chromosomes which we see, and we define them on the basis of the presence of the centromere position. Okay, now chromosomes are divided into two parts, the P arm and the Q arm. Okay, because of the positioning of these centromeres. Centromeres can be located in different positions, and in metacentric chromosomes, they are in the middle. Submetacentric, they are a little off center, so there's a shorter P arm and a longer Q arm. Acrocentric, they are actually severely offset from the center, leading to a very, very short P arm and a longer Q arm. So these are the chromosomes which are involved in Robertsonian translocation. Now, telocentric is where the centromere is actually found at the end of the chromosome, meaning that there is no P arm. So these kind of telocentric chromosomes are not found in humans. So moving on to one of the most confusing questions which everybody has a doubt about, especially when it comes across as a first time. Okay, of course, after discussions, then it becomes a little clearer. So I put it up here for all of you. A woman is 8 weeks pregnant and she's attending genetic counseling. She is known to be a carrier for cystic fibrosis, and her sibling has a child with cystic fibrosis. The carrier rate for cystic fibrosis in the general population is 1 by 25. What is the risk of the child with cystic fibrosis? Is it 1%? Is it 12.5%? Is it 25%, 75%, or 100%? Now, here we have to see what kind of inheritance pattern are we dealing with. And because the partner status is not known, but we are told that the carrier rate for cystic fibrosis in the given population is around 1 in 25. So we will have to consider that as the risk that the partner is also a carrier. And now let's see step by step how to solve this question. So now let's try and solve this question step by step. First thing that we need to know is that the inheritance pattern of cystic fibrosis is an autosomal recessive pattern. So for a child to have cystic fibrosis, they must inherit two defective cystic fibrosis gene, that is one from each parent. Now, we do not know the carrier status of the father. However, we've been given that in the general population, the carrier risk is around 1 in 25. So if that is the risk, then the chance that the father is a carrier is around 1 in 25. Now, if we say that the father is a carrier and we know that the mother is 1, then we draw this Punnett square and we reach a conclusion that if the father is a carrier, then the probability of passing the defective gene from both the parents is 1 in 4. Only 1 in 4 child will inherit both the abnormal genes from either of the parents. So therefore, 1 in 4, that is 25%. Therefore, the total risk of the child having cystic fibrosis will be 1 by 25 into 1 by 4, which is around 1 in 100, which is 1%, and that is our answer here. So I hope that this presentation has been a little helpful to you, and a lot of confusions that are there in your mind should be settled now, and that you will go into the exam a little bit more confident and come back happy with your performance in genetics. So the other important topics that you should be covering are cell cycle, then mitosis and meiosis, okay, the various steps of mitosis and meiosis that happen, the similarities and the differences, the most common aneuploidies and the features that are associated with it, the soft markers for prenatal diagnosis. So they can probably give you the levels, and maybe give you that this is low, the beta HCG is low, the PAP is high, whatever, okay, in various combinations. Then they will ask you what kind of aneuploidy are you dealing with. They can ask you inheritance pattern of various conditions, so you must have a chart and maybe some ready mnemonics, whatever suits you, okay, so which will help you to memorize at one go the kind of autosomal dominant or autosomal recessive conditions or X-linked dominant conditions. So that way you will know how to solve some questions which have a little bit of twist in them. Of course, you will, like we just discussed, the probability questions, that what is the likelihood that the offspring would get affected. Now, I am very comfortable using Punnett squares in that, and really I don't feel the need for kind of memorizing that, okay, 50% will be carriers and 50% will be affected. You are given a rough pad there, okay, so you can scribble, make your own Punnett square or your cross mating, whatever works for you, and reach your analysis as to what the answer will be. So that is in a nutshell about genetics, and wishing you all the best. Please don't stress, and I'm sure you will all do well. Thank you so much, and if you have any questions, please do leave them in the comment box and give us your feedback. And if there's any particular question that you would like to be addressed, let us know and we will definitely get back to you. Thank you.